All right, so I only got one comment back on questions that kids were stuck on when I had put that questionnaire out on the web page. So here's a little video. Hopefully this will help the student who asked about it just kind of understand exactly what's going on. I've got my review book right here. Okay, I marked the pages, I answered the questions for you so that I can try and get through this and explain it in a way that makes sense for you. But anyway, you had asked about question number 54 on page 116, okay? That was the picture of the cell that looks much better than this, but I did it, just fast and dirty so that I can give you your video and kind of explain your answer. So in that picture, you have obviously an animal cell, right? There's no visible cell wall. I should have connected this to show that, but anyhow, you have a couple numbers labeled here. You have one of these tiny little dots labeled with a number 1. You have the large piece in the center labeled with a 2. And then you have that, I told you they love those hot dog looking things, right? With the squiggles in the middle. That's always going to be a mitochondria. Okay, so number 3 is right there, and that's what that's labeled. And they love putting those on your regent, so make sure you know them. Okay? So anyway, 54A says, describe how structures 1 and 2 interact in the process of protein synthesis. Okay? Oh, wow, those numbers are all backwards, aren't they? All right, well, anyway, you can figure it out. But anyway, so structure number 1, that tiny little dot right there, that's going to be a ribosome. Remember, the job of the ribosome is to make proteins, okay? That's its job. It's protein synthesis. It can make RNA, which is, ribonucleic acid, but that's going to be used to make your enzymes. Remember, enzymes are going to be super important in terms of allowing reactions to happen in normal biological conditions, okay? That's what makes it possible for you to digest food and not have a body temperature of 180 to try and break it down, okay? So you have that one. Then you have structure number 2, obviously the big circle in the middle. You've learned it since elementary school. That's always going to be the nucleus, okay? Now, as you know, I always try and come up with a dumb way to remember something so it sticks in here for me. But what I came up with for this one, number 2 is going to be all of your blueprints for everything, okay? That's going to be your head contractor. That's your foreman. That has the list of all the blueprints for everything that has to happen biologically, okay? Number 1 is going to be your ribosomes. Your ribosomes are like your general contractors. They don't need to know every little bit of mechanics going on in the building. They need to know their job, and they need to be able to do that well, okay? So here's your blueprints. That's got the plumbing, the electrical, the structural analysis. That's all your floor plans. That's everything for the building. But number 2 shares just what number 1 needs. I need the plumbing done in this building. Number 1 is the ribosome. Number 1 can make that happen. They take that page, you only need me to do the plumbing, done. And they do that. They only specialize in one thing, and they will make that protein or that enzyme so that the cell can go ahead and function, okay? So that's what's going on with those questions there. Then part B says, describe how structure 3 aids in the process of protein synthesis. Remember, a mitochondria, which is number 3, the mitochondria, right there, is what provides all of the cellular energy, okay? That's involved in the formation of ATP, adenosine triphosphate, ATP. I told you to equate that to cellular gas, okay, or cellular fuel. That's what drives all energy in a cell, is going to be the mitochondria, Okay, so hopefully that helps you answer that one. The next question you had asked me about was on page 144, and it was question number 33. And for that one, this is going to be one of the state labs. Okay, that was going to be next week, but we got ourselves canceled, so that's why I posted the video on there, or on the web page the other day, to try and help you out with these questions. We'll see if that worked. All right, I don't know whether this question still exists for you or not, but anyway, I'll buzz through it. Watch the attached video that I put on. The guy on YouTube does a really good job explaining this. But anyway, you had to fill out the chart, and on that chart it said complete the accompanying table to summarize a change in location of substance C. Let's see if I can get that focused. It's all going to be reversed, of course. But anyway, substance C in the experimental setup. Okay, so the name of substance C, you had, let me see if I can do this just quickly for you. You had the beaker with the little dialysis tubing in the middle. So we go beaker, water, little dialysis tubing hot dog down in there. Ooh, wow, that is why I am not an art teacher, kids, right there. But anyway, and it says you have water and substance C here. So that, I'll try and write it so you guys can see it. Like that. Oh, how do I do it backwards? 2. Oh. Okay. Hey, that actually looks pretty close. The 2's still backwards. Well, all right. But anyway, when you're looking at that, you have the little dialysis tubing hot dog here. Then you have substances A and B and water in here. Okay? So, if it's able to move through dialysis tubing, you know it is very, very small. Dialysis tubing has little microscopic pores in there that allow things to move in and out. So if it can move through dialysis tubing, it's very small. Okay? If it can't, it's like trying to fit an elephant through a screen. It's just not going to work. Okay? And that would be your molecule, let's see, that was molecule B, or substance B in part B. That's your starch. The molecule's too big. It can't fit through there, okay? But the iodine is very small. So the iodine can pass through. If you remember back to last year with Mr. Doyle, you had to do the whole thing with the periodic table and all that, okay? You would remember that iodine is way over, of course you can't see it, I'm just pointing to a wall, but way over on the right-hand side of that periodic table, okay? And with it being all the way over there, that's going to fall into the group that is going to be inert, okay? That would be your iodine, okay? Very, very small, can fit through without a problem. So, for part A, when you fill out that chart, name of substance C is the iodine. That's what turned that water orange or amber. That's how it can pass through the dialysis membrane and react with substance B on the inside of that tubing, okay? So direction of movement would be into the cell or into our model, and the process by which it moves in from an area of high concentration out here to an area of low concentration in here is because of diffusion, right? Things move from an area of high concentration to an area of low concentration. So you got that. That was diffusion. Substance B, far too big to get out, right here. Substance B reacts with iodine. It turns bluish black when you mix the two. So starch can't make it out. Iodine makes it in, turns the inside of this bluish black. Substance B is starch, and the molecule is too large. So that finishes up that question there. Last couple you had, 41 on page 146 in your review book. Question number 41 you asked about. We already did that. That was the one that I went over first, right? You have the nucleus is the blueprint for all the proteins and all of the genetic information in the cell. And then the next one I would put down would be ribosomes. Ribosomes are in charge of building those specific proteins for a specific job, okay? Then you had a question on number 44, and it says, they give you the paragraph, right? Remember, this is going to be a reading test, not a science test, a reading test. On this one, you had to flip back a few pages. It was on page 125 in the book, and it listed it in the reading, okay? So if you read that, it should have triggered something in your head. If it didn't, it says identify the specialized structures in the cell membrane that are involved in communication. Number 5 on page 125 is receptor proteins, okay? Your receptor proteins, they're going to be the ones that connect. If you look back on that drawing, it looked like this on the side of a cell, and it had a few of them. Well, that looks kind of familiar to what we're seeing on the news every night, doesn't it? I'll be darned. It's almost like those proteins stick out around every cell, right? That looks a lot like the COVID virus. But anyway, the receptor proteins, those are going to be what bind to the proteins and allow the reaction to happen. Now it says, explain why chemicals released from one plant species may not cause a response in a different plant species. Well, because the protein doesn't fit, right? It doesn't match up. If I take the wrong key to a lock, it's not going to work. And that's how those things work. Remember, you have the lock and key mechanism that was mentioned in the reading. You have the right protein comes in. Let's say this little protein comes in, lines up, and just docks into that little docking station, and that would trigger a reaction. But if I had another protein, like say that they're mentioned plant species in the article or in the reading, right? So let's say you have a dandelion protein flying through there, and it tries to land on an iris. It's not going to work, right? It's like taking a master lock key and trying to unlock Fort Knox. It's not going to do it, okay? Question number 45. They showed you a drawing that looked kind of similar to that, but they, of course, have to change it just to mess with you a little. So I'm going to make this fast so I don't get this video too long for you. But anyway, you had the beaker filled with water. You have a test tube. The test tube was like this, and it had the dialysis membrane on the bottom this time, but it's still going to do the same job. It's only going to let small things through, not let big things through. And then you're going to have the starch and water inside this time. So starch plus water on the inside of the mixture. And then you have, they had to make it fancy. So they put a ring stand here holding onto the test tube just to try and mess with you. Okay? Explain the procedure used. All right. So number 1, you put the starch and the water inside the test tube. Okay? Then you put dialysis membrane right here. Then you invert the test tube and you put it in the ring stand. Bloop! Right into that beaker of water. Okay? Now you have a water beaker, and it says right in the directions, where was it? A laboratory setup for the demonstration represented in the setup. An indicator can be used to determine if starch diffuses through the membrane into the beaker. So what would you do? Well, you would just take a drop of iodine or 2, drop them into the water here, and if it turns bluish black, you know the starch is diffusing from in here to out here. Okay? Since that will not happen because starch is far too big to fit through dialysis tubing, it can't make it, so that's where that one stops. All right? If the water outside of the test tube changes color, you will know that the molecule was able to make it through. And then it ends with part C. Identify one type or another type of organic compound that can be identified with an indicator using this type of laboratory equipment. If you were to heat it up, you could check for glucose. Okay. The last one that I just got to buzz through here, so it's not a half-hour video, was question number 48 on page 148. You had a question on that. They give you the little equation where it says digestive starches in the bloodstream. They they go to a cell, then a cell structure produces ATP, okay, with the arrow going to ATP. All right. Explain what occurs beginning with the digestion of starch and ending with ATP production in your answer. All right, so step A says identify the molecules that are used to digest the starch. What do you use for digestion? Enzymes. Enzymes facilitate digestion, okay? So it's the enzymes that are doing the work. Then it says identify the molecules produced when starch is digested. That was back in the previous chapter too. That was about the chemistry, okay? And you take a large cell and you break it down, that is digestion. If you take a small cell and you put it together to make a large one, or a small structure and you put it together and make a large one, that is synthesis. What does starch become? A simple sugar. You put together enough simple sugars, you're going to make yourself a carbohydrate, okay? Or I'm sorry, you're going to be making yourself a starch. They're both carbohydrates. It's just one's simple, one's more complex. Explain why the starch must be digested before building block molecules can enter into the bloodstream. You suppose this might be important since this is the third question that they've beaten it into your head on? Yeah. Why do you have to break down starch? Because starch is too big to pass through the membranes. Okay? Then you get to letter D. Identify the structure in the cell that will produce ATP. Again, that ties right back to the first question that we did to begin with. What makes ATP? The mitochondria. That little oval-looking thing with the squiggles. Okay? That's the mitochondria. That makes cellular fuel. That makes the ATP. That makes the energy. Okay? All right. And I didn't mean to, but that actually answered the next question where it says state why ATP is important. It's important because it's cellular fuel. Without ATP, you don't have enough energy to get out of bed in the morning. All right? We'll talk to, and your heart can't beat and your diaphragm can't work and all the other cool stuff your body can do. Anyway, thanks for watching. Hope that helps.